We'll be live in 12 seconds. Are you able to share the slides? I'm not seeing the screen yet.
I'm trying to. All right.
Hi, everyone. Thank you for joining this session where Nader Hamzavi will talk about the quantitative systems toxicology models for drug-induced renal injury. Before he gets started, a couple of housekeeping remarks. We take your privacy seriously by attending this event or participating in the Q&A session. You are allowing us to contact you for follow-up. You may ask questions via the Q&A tab on your dashboard at any time during this presentation. If you need technical assistance, please use the chat tab to raise your questions. And just a very quick poll before he gets started, just to see what kind of audience we are talking to at this presentation. What is your area of expertise? Engineering, mathematics, pharmacometrics, systems biology, pharmacology. And we'll wait another 20 seconds or so before closing this poll. And hope everybody managed to submit your answers. We do have nearly 60% of our audience today is coming from the pharmacometrics field, 35% from engineering and mathematics, and the rest from systems biology. So I guess a lot of the audience is here to learn about the systems biology models. So go ahead, Nader.
Yeah, thank you, Vera. My name is Nader Hamzavi. I'm Scientist 1 with Diligent Services, a division of Simulations Plus. So this poster presents our efforts to develop a quantitative toxicology model for a prediction of acute kidney injury. And we were looking at its association with renal hemodynamics. So first, we look at this special type of cells, kidney cells called proximal tubular cells, since they are at the forefront of the exposure to the drugs and they are vulnerable to drug-induced toxicity, which often leads to, I think 20 to 30% often leads to acute kidney injury. And this injury varies from mild to severe injury, and in some cases leading to cellular death in the form of necrosis or apoptosis. And at the systemic level, then the AKI manifestation is decline in filtration rate. It's that we call it GFR as a glomerular filtration rate. So now the challenge is how to predict AKI, how to have a reliable prediction, considering all the complexities related to pathophysiologies involved, like cellular, hormonal, and hemodynamics. And so our approach was using quantitative systems toxicology, trying to use it as a quantitative and computational tool to translate in vitro mechanism data to renal function and renal hemodynamics. So this project has resulted in releasing the first version of Renasim, the platform for QST modeling of kidney disease, acute kidney injury, and it has been It has been a team effort sponsored by SBIR and IDDK, grants, and people. There are a huge number of our team working on this software development. And I'm going to talk about the methods that we use of this software, the multi-scale approach that we did, and we are going to look at the cisplatin representations in RENASYM and some of the results that we have already gathered. So at the cellular level, RENASYM is actually representing the life cycle of proximal tubular cells, how the cells are going from young to mature and how they are regenerating. We're also including the bioenergetics, immune responses to renal toxicity. And from the in vitro assays, we are gathering information and we are parameterizing the key cellular injury mechanisms that I will talk to it in a few seconds. But at the systems level, then we are also representing the renal function and the feedback mechanism. This includes the tubular glomerular feedback, renin, angiotensin, aldosterone systems to regulate the blood pressure, the water, and accounting for sodium reabsorption at the cellular level. So the model is actually integrating the cellular injury to the systemic level, and to predict the injury to the proximal tubular cells in humans and also in rats. So I'm presenting the results that we have for rats, and we have more data on rats, and we could use those to validate the model. So as you can see in here, we have the schematic of all the compartments that we have in RENASYM. For the RENASYM representation of cisplatin, cisplatin is a chemotherapeutic drug which has been effective, but it has side effects on normal tissues, and one of the dose limiting factors is the side effect on kidney and nephrotoxicity. So we started with building a PBPK model for cisplatin in GastroPlus to estimate the drug exposure that's getting to the cells. And then similar to other projects that we are doing in DILISYM, we gathered in vitro measurements of two major mechanisms that is represented in RENASYM. One is oxidative stress, shown in here, that is generating oxygen consumption rate, and we are using a third party to gather those in vitro measurements for us using high content screening method, and telling us how much of the reacting oxygen species are generated. They're also gathering information about mitochondrial dysfunction as the second toxicity mechanisms included in here by looking at using Seahorse XF analyzer and looking at the oxygen consumption rate. The other mechanisms in here as well, like crystal nephropathy, but it is not relevant to cisplatin. So using those in vitro information, then we can parameterize our toxicity measurement, toxicity mechanisms, and that will lead to the effects on the cell life cycle, and the cellular health will be affected by those, and that is going to affect the biomarkers. So we have two set of biomarkers in RENASYMs. To general case, one are looking at the function, one are functional biomarkers, looking at the systemic effects of the drugs, and one is urinary biomarkers like alpha GST or KIM1, which are representing the cellular injury. But if I go to the results sections, and so Here we are looking at a rat cisplatin model and at two different doses because it is hard to find all the information at one dose, and we want to also test the model at multiple dosing as well. So the first thing that we did is looking at the drug concentration and comparing the PBPK simulation results with the data. Then we are looking at the plasma concentration as well as the kidney concentration in here. That's also important in slow clearance of cisplatin from proximal cells, proximal tubular cells. Knowing, getting the pre-exposure correctly, then we can go to the cellular responses. So we have some information in here for reaction oxygen species from the data and our simulation results. Now you can see that our predictions or the model is actually optimized to recapitulate those data pretty well for ROS as well as ATP decrement. And that's defining how much cells we get, how much cells we lose due to the drug concentration, to the cisplatin concentration. It is hard to find exactly the information about like the PTC loss, the number of cells or the number of nephrons, but we have some information about the tubular cells with necrotic cells. We use them as a marker for cell viability. And you can see at the higher dose, we are losing like about 40% of the nephrons based on our predictions. And this is, in a way, we are considering that 5 mg per kg is a high, it is a severe dose, so we expect that to happen. But when we go to a lower dose that we don't have more information about ROS or ATP, but we have information on biomarkers that we are using here for validation. So if you look at our results at the lower dose, then you can see that the recovery is actually faster. We would say those cells are fully recovered, and the filtration rate is also recovered. And you can also see that serum creatinine is also peaking around day 5, which is in agreement with the observation, although the magnitude it's needs to be improved maybe. And we're going to address that in later versions of the software. And we also look at the urinary alpha GST, which is a marker of necrotic cells. We could capture that as well. And also we have the urinary Q1, which is a marker for which is peaking after the peak of maximum injury. And then when the cells starting to recover, starts to slowly going down. And we could capture those biomarkers with this system. So if I wanted to conclude, we could evaluate the cisplatin-mediated toxicity, dose-dependent in rats, using Renasim, and we also it shows that Renasim is a promising tool to actually predict AKI and evaluate preclinical as well as the clinical outcome that we are working on, not only for cisplatin, but we are also working on other nephrotoxic drugs also included in Renasim, first version of Renasim. So I can stop here for questions.
Yep. Thank you, Nader, for a great presentation. I know this wasn't nearly enough time to go over all the details of the model. Unfortunately, we are running out of time, so we won't be able to address any questions. But for those of you who have questions, I'm sure that you can reach out to Nader directly, and he'll be happy to discuss this with you. So thank you all for joining us. This webinar has been recorded for playback and will be available on our website or YouTube channel. And this concludes the session. Please head over to the lobby for the next presentation. And don't forget to join us for the panel discussion that will follow the next presentation in this preclinical track.